For the past few days in class, we've been looking at situations that don't fit the Mendelian inheritance pattern. This one is so big of a difference that it deserves its own video. This is video 11.4 on linked genes. This video will cover these two goals. We'll talk about what linked genes are, and then look at some genetic data to see how you can identify situations when genes are linked. Linked genes were first described using a fruit fly model, so let's dive into linked genes by looking at this classic experiment. In this experiment, we'll follow Drosophila body color and wing shape. There are a couple of things you need to know before we start. First, this video will use the term wild type. Wild type just means the normal phenotype or a non-mutated phenotype. Another thing is that in Drosophila, they name a lot of the genes after the phenotype that's caused when the gene is mutated. The example in this experiment is vestigial wings. We've heard of vestigial structures before, like how whales have vestigial hips. Vestigial structures are structures that are there, but not really useful, just like these vestigial wings. In this case, you can see that the vestigial winged flies have these tiny, non-functioning wings. All right, so let's sink our teeth into this. In Drosophila, gray body and normal wings are both completely dominant to black body and vestigial wings. A purebred, wild-type fly is bred with a purebred, black-bodied vestigial winged fly. One F1 dihybrid fly is bred with a black vestigial winged fly, a double mutant. Predict the phenotypic ratios of the F2 offspring. If you consider the sperm and the eggs that are made by these F1 breeders, you can predict what the F2 offspring will look like. With independent assortment, this female can make 4 different types of eggs. Since the male is a double mutant, he can only make this one type of gamete. When you cross these up, this gives us an expected ratio of 1 to 1 to 1 to 1, with equal amounts of each phenotype. This experiment, in reality, however, gave some unexpected results. Here's the data found in the original experiment. There weren't the same number of each phenotype. The results were skewed so that the original parental phenotypes stayed together. Wild-type gray body and wild-type full wings, mutant black body and mutant vestigial wings. You can figure out the ratio, because these big numbers are kind of confusing, just by dividing all of the numbers by the lowest number, 185. Doing that gives us the ratio of about 5 to 5 to 1 to 1. Once again, scientists were confronted with something that didn't fit Mendelian inheritance. So they had to have some imagination and rethink how inheritance works for cases like this. What they found was that not every trait assorts independently. They found an exception to this rule. Independent assortment is actually a rule that applies to whole chromosomes and not individual traits or genes. If the loci for a trait are on the same chromosome, then those loci will assort together and not follow independent assortment. Thomas Hunt Morgan was the first guy to describe this in his flies. That's the same Thomas Hunt Morgan that also described sex-linked genes. So what he came up with was the term linked genes to describe genes that are on the same chromosome, especially when those genes are close together. When genes are linked, the genes both end up in the same gamete during meiosis. Let's look at this example over here. We're following 3 loci, A, B, and D. Since A and B are on the same chromosome, A and B are linked. Since B is on a separate chromosome to D, and A is separate than D, and D is not linked to A or B. It's easy to imagine how this would play out during meiosis. This diagram shows all of the possibilities of that ABD cell on the last slide. Chromosome 1 and chromosome 2 assort independently. During metaphase 1, You can have both blue chromosomes end up on the same side of the metaphase plate, and both pinks end up on the same side, or one pink and one blue on either side of the metaphase plate. If we follow this through the rest of meiosis, you can see the result of gene linkage. The bottom shows all of the possible combinations. Little a and little b always stay linked together. Big A and big B always stay linked together. Only locus D assorts independently of A and B. So how can you tell if genes are linked? Well, you need a lot of data from a lot of offspring for the patterns to emerge. But it comes down to comparing the expected ratios of Mendelian inheritance to observed ratios. This is starting to sound familiar? It should. All right, let's check this out. Here's the data from Thomas Hunt Morgan's fly experiment. If we expect the numbers of each phenotype to be equal, that makes 575 of each. Now with the data arranged like this, you can do a chi-square analysis. I'll save you the trouble. The calculated chi-square value would be 1,002, a huge number. So in other words, there's a statistically significant difference when you compare the inheritance of these two fly traits, body color and wing type, to a model of Mendelian inheritance. That difference is not random, but scientists have explained the difference by the genes being on the same chromosome. But there's something else that's weird about this data. Gray body and normal wings don't always stay together, just like black body and vestigial wings don't always stay together. So there's something else going on here. The linkage between the two traits isn't a complete linkage. So this begs the question, when can genes unlink? Well, the answer lies in our knowledge of meiosis, specifically prophase 1 of meiosis. Remember homologous recombination, crossing over? That's when homologous chromosomes pair up and swap equal pieces of DNA in prophase 1. If the crossing over swap happens between the two loci on that chromosome, then these loci will become unlinked. You can see the unlinking of A and B in this diagram, where the crossing over happens between them, and now big A is paired with little b, and little a is paired with big B. Loci A and B are linked because they're on the same chromosome, but when crossing over happens at any point between loci A and B, then the loci become unlinked. There's a term for this called a recombinant chromosome. A recombinant chromosome is one that causes a recombination of the linked loci. So these two are recombinant chromosomes. If the chromosome is from the side that didn't do crossing over, or if the crossing over didn't happen between the two loci, then it's called a non-recombinant chromosome. And you can see those two over here. If the crossing over was just a little nub at the end, we'd call that a non-recombinant chromosome in terms of unlinking A and B also. One other thing while we're here, imagine if A and B were closer together. If that were the case, it would be less likely that this crossing over, the break between the DNA, could unlink the A and B loci. So the closer the genes are, the more linked the traits will be. Before DNA sequencing came into play, scientists actually determined how far apart genes are by looking at how frequently the genes recombined. Check out this example. Loci A, B, and C are all linked genes because they're on the same chromosome. But since A and B are so close together, it's less likely that there will be a crossing over that recombines A and B. It's way more likely that crossing over will separate C from A and B. So, what we could say is that A and B are more tightly linked than either of those are with C. I want to leave you with one last thought. A lot of people think Mendel might have kind of cheated, or at least was working with bias. Mendel's data was really good. A lot of people would argue that it was too good. Look at how close all of these ratios are to the expected ratio of 3 to 1. Did Mendel fudge some data? Also, for all 7 of these loci to behave the way they did, they'd all have to be unlinked, and they'd all have to follow Mendelian inheritance. What are the chances? With so many exceptions to Mendelian inheritance, how is it possible that Mendel picked 7 traits that all happen to follow this inheritance pattern and gave such clean data? The idea that Mendel's data might not be as pure as what we'd like is called the Mendelian paradox. You can read about it more if you Google it, and it's pretty interesting. But how could we call a frickin' monk a liar? That's kind of crazy. All right, so what did we learn in this video? Let's recap. We learned about linked genes. Linked genes are genes that tend to stay together in gametes and are seen together as traits in the offspring. The reason why linked genes happen is because their loci are on the same chromosome. But linked genes can become unlinked during crossing over in prophase 1 of meiosis, when crossing over forms recombinant chromosomes. Finally, linked genes are not only on the same chromosome, but how close they are together determines how linked the genes are. The closer the loci on a chromosome, the more linked the genes will be.